Welcome everybody to the NextGen Microfluidics webinar. This is the second in our series of demo case webinars from the H2020 project NextGen Microfluidics. We're happy that you're here and we look forward to presenting our speakers. I'm going to hand over to our coordinator, Martin Smolka. We'd like to ask everybody to mute yourselves and turn off your cameras during the presentations, as this will be recorded. At the end for question and answer, we're happy if you unmute, raise your hand, and ask questions. So, Martin, I'll hand over to you.
Thank you very much, Caitlin. So, first of all, you here see the intro slide about today's topic. And all in all, we will go through the following points. So, I am first of all giving you an overview over NextGen Microfluidics, over our EU project, and then I try to be very short and hand over to George and to Jörg. So the main thing, so when we started NextGen Microfluidics, we saw a big challenge in microfluidics industry in the development of microfluidic devices. And as you know, this topic is rather complex. And typically, so you need to develop separate part of your microfluidic chips and often need different players for that. So different components also need to be developed by different companies, by different solution providers. Therefore, a customer or somebody who wants to get chips developed needs to choose between different players, needs to verify that all technologies are matching. So there's a lot of work for an innovator, for example, a startup with a new microfluidic technology who wants to get the chips produced. What we therefore did in NextGen Microfluidics, here you see our logos. What we did in NextGen Microfluidics, so first of all, we acquired quite a lot of money. So it's a 17 million EU budgeted EU project. And we collected 21 solution providers, which contribute to the development and production of microfluidic devices. So they contribute at different points of the value chain. And so we cover all the aspects starting from design to final production. We all together, we got the chance to found our own startup, the Microfluidics Innovation Hub. And now, so external customers can address this Microfluidics Innovation Hub, and it combines all the services which we are offering and gives you a combined service portfolio. So we have one single entry point which a customer can address. And so with this setup, we already did quite a lot of verifications of our capabilities, of our technology portfolio and our service portfolio. So at the beginning, we had specific applications already defined. So with our portfolio, we worked on 5 predefined microfluidic applications. And then with these first verifications, we went out to the world and acquired external customer projects. So we got requests from external customers and worked on their application. And all in all, we demonstrated our portfolio with 22 applications so far. So all in all, that's a rather market near project. And yeah, the final slide from my side. So with all that, we are contributing to the microfluidics revolution in a leading role and cover quite a lot of technology steps. So the part of how you get the chips, how the chips are done, how you integrate sensor, biosensor functionality, and this requires expertise in a broad range of technologies like chemical, biochemical, and biological know-how, so that we can really cover the broad range which typically you find in microfluidic applications. So lab-on-a-chip, point of care, in vitro diagnostics, but also chips in which you grow cells, so organ-on-a-chip applications. And with that, whenever you you're asking yourself how to scale up your application, how to get microfluidic chips produced and developed, then please contact the Microfluidics Innovation Hub. Thank you very much, and we are looking forward to hear more from George and from Jörg. And sorry, Caitlin, short advertisement. We organize a small microfluidics conference. So there is a medical technology event in Graz, here where we, the coordinators, are situated. So it's the Health Tech Hubsteria, and a special session on the second day, the 26th of March, is dedicated to microfluidics, and we are happy to see you there at the end of March. Here, all our partners, and with that, I really hand over.
Great. Thank you so much, Martin, for presenting the NextGen project and Microfluidics Innovation Hub. I put some links, some helpful links into the chat for the participants. And now we'll hand over to our demo case leaders, George Sikanis from BRFAA and Jörg Nestler from Biflow Systems.
Okay, so today's talk is about lab-on-chip technology and also the biochemistry put on lab-on-a-chip systems. And as the NGM project is also a lot about manufacturing, I'd like to cover some aspects of manufacturing today and things to keep in mind when it comes to really make a product out what was developed. By the way, what you can see here on the left side, I hope you can see the video, is a cartridge in a cartridge holder, everything which runs on that cartridge. If you can see the liquids moving here at the bottom side, this is done by integrated actuators. Just some quick words about us and our company. So we focus on, as you can imagine, lab-on-chip development and manufacturing. And the special thing of our technology is that we can integrate all the functionality which is typically required, like moving liquids, like heating liquids, and that's all integrated on the cartridge. And of course, the reagents. Just to cover a little bit the applications we were targeting in NGM project, one of the applications was SARS-CoV-2 and influenza discrimination. And what we did is we used one of our cartridges and added a microarray. Everything is done by partners in the project, and this microarray held then also the spots of different target molecules. Now, what you can see on the left bottom side is the spotting equipment at Joanneum Research, which is a roll-to-roll spotting equipment. And here you can see some results done with an instrument you can see on the left side. And this is actually a microarray pattern after the hybridization of a PCR on a cartridge. I'll give you another example, which is not part of NGM project. It's a completely different aspect, but done on a similar technology, similar cartridge technology, which you can see here, which is the identification of allergies, which is ideal as an application for a microarray-based solution and also an integrated solution. Because as you can imagine, for allergies, you need to look at a lot of parameters, a lot of allergens at the same time. And for that, running a microarray is ideal. And this has been done here as an example for 13 different allergens, or allergen components. But of course, it can be extended easily to several hundred. Now, today, I more want to talk about disposables as such and the manufacturing of that. And when we talk about disposables today, I'm not talking about lateral flow strips, as you can see on the left side, but more on the more complex solutions, as you can see on the right side. And you see some commercial examples, some of them even don't exist anymore, but they were quite advanced or are quite advanced, and there are a couple of them out there, and even after COVID, there are more of them. And this is the type of disposables I would like to talk today. And if it comes to such disposables, you will always have raised the question of the level of integration. Of course, you can always argue, and people are arguing, that there should be low level of integration because typically it gives you a much cheaper disposable, but on the other hand, normally it makes your instrument more complex, you may have more effort on maintenance and things like that. And even the size of the instrument may become larger. If you look to the high level of integration, most likely the disposable is more costly. And people like cheap products, as we could also see in COVID times. Everybody was screaming for European, or in Germany, German solutions for lateral flow strips, but after they were available from China at lower cost, nobody was interested in them anymore. So yes, cost is an important point, always. The advantage of integration, however, is that you can easily control the disposable without complex like pumping mechanisms, mechanical mechanisms, and so on. So reliability of the instrument is an important point here. And of course, also maintenance, because you don't have reagents in the instrument, for example. And when it comes to operation by laypersons, especially for complex tests, so not talking about things which you can easily do by a lateral flow strip, then you may end up with a more complex disposable, simply to make it easier to be operated by the people. And I'll give you an example going back to this slide. On the left side, actually, you see, I would say, quite cheap disposable for an allergy test. Now, what you do not see here for that allergy test, or you see it actually, is these are the numbers of vials and reagents you need to pipette one after the other to that cartridge. And actually, it takes you 45 minutes hands-on time to have a point of care solution for allergy diagnostics for 20 parameters. While with a cartridge like that, it takes you like 1 minute sample in, putting the cartridge in the instrument and waiting for the result. So this is the disadvantage of the low level of integration here, which makes it less ideal for a couple of point of care solutions. Okay, but at the end, it depends, as you can imagine, on the application and the use case. There is also an issue, now we come to manufacturing, when it comes to quality. The more complex disposable becomes, the more issues you have with quality control. Assume you have a process of manufacturing of 20 process steps with a yield of 98% for each process step. The final yield would be 67% only. So you lose 33% simply because yield and 98% is not too bad. Now, imagine you have 50 processes. By the way, 20 sounds a lot, but if you look into the details even of simple disposable, 20 is not too much. If you look to 50 process steps, you end up with 36% yield. So this is really a problem. And if the actual quality outcome, the yield for each process step for 20 process steps is just 95%, it's also 36% final yield. So this is really an issue why you actually have to carefully think about complexity of a disposable. And even more, a quality issue in a single disposable, most often you cannot easily detect in the final QC. So you have to do the inspections for all the process steps. That's also something to consider. And at the end, it's important that you need 100% in-process quality controls here. And this basically increases the cost of manufacturing a lot. So at least our experience, and we also talked to a lot of other manufacturers, you can easily estimate that 50% of your production costs are for quality control. And this is typically underestimated. Yeah? So whenever you develop something like that, keep that in mind. Now, speaking of the number of process steps, there is a value chain for these kind of disposables. Of course, you have the assay, and you will see a slide like this later, most probably as well, because this is the value chain of Microfluidics Innovation Hub, developed in the NextGen Microfluidics project. So you have an assay which is integrated on the disposable. You have a design, which is part of the value chain of development, but not of the production. You have the patterning of substrates, can be injection molding, can be embossing, can be roll to roll, or mixture of everything. You typically need to functionalize at least one surface, and if it is by capture molecules. Sometimes you even have electrodes, at least you have whatever kind of sensor. Then you need some back-end services, and this is also underestimated. You have bonding involved, you have sometimes cutting involved, so different steps. You have packaging involved, so different steps which come after actual manufacturing of single parts or the final part. And then at the end, you need whatever kind of device to do readout, and in some cases, the control of the disposable, and for everything you need quality management. So this is the actually value chain of Microfluidics Innovation Hub, and looking into what is related to the disposable. You see, you have the assay, you have the patterning, surface functionalization. If there are electrodes, you have the electrodes and or the sensors, and you have the backend. Yeah, and the point here is that this is quite complex. A large number of different sciences, different competencies is required here. And I would say that there are not many companies around, even also not many research institutes which cover everything. I would say only the very big players can really walk alone here. And even some of them didn't go for walking alone. They partnered with others. And it's also the approach of Microfluidics Innovation Hub. So we have the companies and the institutes to cover different aspects of what is needed in the different parts here. So not only covering from left to right, but also within all the different aspects, we have plenty of solutions available. So whatever kind of solution is required to solve a problem, we can look for it internally. And if we don't have it internally, we can look for it outside. And speaking of only big players can walk alone, not even all of them succeeded with that. So this is something which was introduced already. And as I said, you cannot have a single solution. So, for example, you cannot only go with injection molding when it comes to structuring of substrates. You cannot only have a solution like roll-to-roll manufacturing when it comes to structuring of substrate. You need to look in the portfolio of available technologies to fit to the applications. And speaking of experience and speaking of what has been done in the different or open core projects in Microfluidics Innovation Hub and NextGen Microfluidics. Actually, this was the way it was done. So some of the applications required glass, some of the applications required polymers, some required roll-to-roll manufacturing, some others injection molding. Assay-wise, it's also completely different aspects were here covered. So that's why having a one-stop shop, not only having its own technologies, but really looking what fits the problem best. This is something which MIH, Microfluidics Innovation Hub, can do. And I have a second last comment on the manufacturing of lab-on-a-chip disposable. So we are often confronted with customers asking for development of a point of care product, and they typically ask, okay, we need 10 million pieces per year after 3 years, and of course everything has to be below €1 and so on. Okay, but reality, to our experience, and we're in the business now since almost 15 years, is like, yeah, you start with a couple of hundreds pieces, maybe 10,000, 20,000 pieces, 20,000 maybe after 5 years, and the 1 million pieces may come later when things really enter market, and sometimes this 1 million pieces never come, and definitely everything is beyond €1. So that's the reality we saw so far, and to keep that in mind, both from a customer perspective and also from a developer's perspective. Now, as we talk about numbers, speaking about scaling of production, this is an example from our own production. And so normally you would not start with an automatized line, yeah, because you can pay several hundred millions and then finally realize that maybe required a slight change in the technology for whatever reason when you do this too early. So typically it's starting with a manual but well-established production, including all the quality control steps required. And this is then transferred to pilot line, automatic pilot line, in this case for our own products. And this is then transferred to a large-scale line for these several million pieces when the customer actually really runs into the market. And then some final remark on where the money is earned with that disposables. Now here you see it's not the latest version of market study, but it gives you the trend. So what you see here is actually the market value, revenue, if you would like so, for two different things. One is going up to whatever 700 something million dollars. The other one is in the $10 billion ranges. Now guess what the bottom and the top is. The top is the diagnostic test. So this is where the money is earned. The bottom is the device. So that's the hardware platform actually the thing runs on. So whenever developing these kind of products, also keep that in mind. And with these initial remarks on the manufacturing of lab-on-chip disposables, I may hand over to George, who gives you some more insight on some of the aspects, especially regarding assay development, which was done in the demo case, but maybe also on some cartridge-related aspects and the whole value chain. Thank you.
All right, thank you so much, Jörg, for stepping in and going a bit out of order, but for sharing your really nice slides. Do we have any questions at this point for Jörg before we hear the rest? If not, you can save your questions. We'll try one more time the screen sharing from George, and if not, then I will share the slides from my screen.
Okay, so this is not the actual presentation, but I will use it as this is a previous presentation. I will use the information shown to actually exemplify what the demo case that we're discussing was all about, and how we brought all of the deliverables that we had to achieve to successful completion. And I mean, the main thing that we wanted is to actually, and Jörg has done a fantastic job in stealing my job in presenting a lot of things, was to develop what you see right in front of you, right? Which is a lab-on-a-chip or a lab-on-a-cartridge system, which is versatile and can be implemented in many different scenarios and applications. And but at the same time being compact enough and bringing together certain innovations that keep the cost low, while at the same time being versatile so that it accommodates different functionalities and all the different processes that are required depending on the type of assay one wants to perform and the type of analysis. So these innovations could be summed up in these four bullets you see radiating from the cartridge, this generalized cartridge design that you see in front of you. And they have to do with the development of the assays themselves, which were customized and custom optimized for the specific cartridge. The cartridge actuation, which Jörg has already presented, and I think I've got some slides in this presentation coming up. The sensor signal readout. Now, this is an interesting one because the readout combines. It happens on the sensor foil, which is, I think Jörg showed you, it's a very thin layer of thermoplast, which is fabricated by roll-to-roll techniques. And that helps not only to bring the cost down, but also to introduce some structures to this really thin foil. So this foil is basically where we spot probes specific to the assay that we want to run. And therefore, this spotted microarray can combine spots for different sorts of analytes. So thinking about molecular diagnostics, as Jörg said, these spots were employed for the differential detection between COVID-19 and influenza A, or the strain differentiation between H1N1 and H3N2, two different strains of influenza A, all in this really small area. Now you can't see my cursor because I'm not sharing the screen, but the area which is in between this white flap at the end of the cartridge, that's the sensor foil, and the white area kind of marks. Yes, thanks, Caitlin. Marks the area where the chamber, basically, through which anything that has been processed, genetic material that has been pre-amplified on cartridge, flows through and above the spotted microarray and hybridizes with probes specific to different sorts of analytes, different genes when it comes to molecular diagnostics, and therefore allows for the multiplex detection of lots of different things in a very small area. Now, this sensor foil not only is produced by roll-to-roll techniques, but as I told you, it bears some structures on it, which allow for the actual detection of the signal generated once we have the material that has been processed, amplified, whatever the test is, running above the sensor itself. So the principle of operation is based on the detection of changes in fluorescence intensity, but unlike going for normal epifluorescence, which is like illuminating the surface from the top with a light beam at a specific wavelength and collecting the emission at another wavelength, we work with TIRF. TIRF stands for Total Internal Reflectance Fluorescence, and it's a variation of fluorescence, which is very interesting because it helps you reduce tremendously the background that normal epifluorescence has. How do you achieve that? The illumination of the sample and of any fluorophores that are to be found in these specific spots happens not from the top, but it's more like the evanescent wave which propagates along the sensor foil itself. So these structures that are at the two ends of the sensor foil, spanning the sensor, the chamber that you see there, actually allowed light to get coupled in the sensor foil, which doubles up both as the surface onto which the biochemical recognition of the material passing above the sensor takes place, but also as the sensing element for the signal detection, pickup, acquisition, and transduction into, with the use of a camera into a measurable, quantifiable signal. So that's how we achieve the reduction, the great reduction in the background noise that we have, simply because we only detect we pick up the signal from a very small area above the sensor foil itself. So in this small area of the cartridge, you can see how many innovations get combined. The actual probes themselves are very specific and selective to specific analytes, and they allow, when it comes to molecular diagnostics, the differentiation between H1N1, H1, H3N2. This is just one assay that we use to illustrate the capabilities of the cartridge itself, but also the detection of different viruses, or in the case of food, the use of atoms. Perhaps we can, Caitlin, if you move. Yeah, it says wait. The next slide. Yeah, there you go. If you go full presentation mode, you will actually see what I was talking about. Perfect. Okay. A bit
Is that too
further
far?
back. No. It's like when you first change the slide from the previous one, where the cartridge and the innovations that are combined are shown. Yes, exactly here. Hang on.
Yeah, not too bad. Okay,
Yes.
I'll get there.
Okay. Sorry. Just press. Yeah. Okay. It's yeah. So this is the first UK. I mean, I'm gonna start. I've already explained this, but this is a graphic illustration of these two different scenarios, these two different applications where the sensors and the cartridge, the lab-on-a-cartridge that was developed and its capabilities was actually validated. Right. The first one has to do with this molecular diagnostics for public health monitoring and the detection of these two viruses, as well as the differentiation between the two different strains of influenza. So there, what you actually see here is the protocol itself, which, as I said, it was custom optimized for this specific lab-on-a-cartridge, or if you want better still, for lab-on-a-cartridge devices. In what sense? In the sense that, yes, we do go for the amplification of the genetic material of the virus if present prior to its detection. There's no secret there. But unlike what is normally done on lab-on-a-cartridge devices or portable biosensors, we don't go for the isothermal techniques, which are the ones usually used in these sort of setups, exactly because of the problems that they have when it comes to specificity, sensitivity, multiplexity, and so on. Here, what you see is normal PCR, however, with a lot of twists. So one of the things that one of the main innovations, for example, that we introduced is that we did employ PCR, but unlike normal PCR, we changed the concentrations of the primers employed to amplify specific genes, so that we amplify differentially and preferentially, if you want, one of the two strands of the genetic material that we were looking for. That already provided us, along with some techniques to actually optimize the procedure, provide us with ready-made single-stranded genetic material that could hybridize with this spotted microarray without the need to introduce a step to dissociate the two strands. So no need to heat, no need to chemically dissociate the strands, ready for hybridization with a spotted microarray, which I don't know why this is like that, but that was meant to, this thing that is standing up, the gray thing along with the spots, was meant to spotted microarray in the hybridization. Now, Caitlin, if you press once more. Yeah. So the other use case, yeah. Yes. Okay. So in the other use case, we used the exact same setup, the exact same cartridge, the microarray detection, with a completely different application. Now, in this scenario, we had to work with milk and develop a device, a cartridge, that uses aptamers for the specific and selective detection of two different classes of contaminants commonly found in milk. So basically to validate the performance of the fabricated cartridge for food safety and quality monitoring. Okay, so the contaminants I'm talking about were aflatoxin M1, one of the major contaminants in milk, and antibiotics. And the first thing that needed to be done was to develop appropriate protocols to extract these analytes from milk. So a lot of work was invested in developing these protocols and trying to integrate them, making them as field amenable, friendly to be implemented in the field as possible. And in the ideal scenario of getting integrated within the cartridge. That would be the holy grail of sample pretreatment for lab-on-a-cartridge. We were not able to achieve that, but since this work is ongoing with some new projects on it, we are hoping to integrate at least as many parts of it, of this sample pretreatment as possible, on cartridge and finding solutions, because this is, as I said, one of the most challenging areas when it comes to analyte detection, contaminant detection in different food matrices, right? So moving on to the actual detection of the extract, which, as I said, the extraction did take place in our case of cartridge, we employed aptamers. So Caitlin, if you move one more slide. Yeah, this shows you that the actual detection was identical. Again, the spotted microarray, but this time the probes that were specific to the contaminants were aptamers. So okay. So again, in the presentation that I had prepared for today, the aptamers and the aptamer-based detection was presented in a more general form, unlike here. But the main point being is that aptamers, for those of you that are not aware of what they're the counterparts, if you want, of antibodies. However, antibodies, unlike antibodies, they have a lot of advantages, such as their significantly lower cost, ease of production, and modifications, all of which allows us to create a biofunctionalized surface capable of detecting with the same sort of selectivity that antibodies detect specific targets, but a much more robust assay in that it has low requirements when it comes to storage. Aptamers, being nucleic acids, do not have the same requirements, and they're not so sensitive as antibodies when it comes to storage, or the fact that they can be easily and site directly modified allows us to really immobilize them on the surface in such a way so that we achieve a really sensitive assay for the detection of these contaminants. But first thing first, the problem with aptamers is that there are a lot of sequences out there which some of them don't work at all, or some of them have really bad affinities when it comes to the interaction with their target analyte. So what we did within the project is developed a process to actually screen the published sequences as well as some engineered by us sequences in order to identify the best performing one. The sequence that has the best binding affinity for a target analyte, the best selectivity for this specific analyte versus other structural similar analytes. So I don't know if you can press. I'm not going to try to even explain what you see here. These are just results. So for example, these are all sequences, aptamer sequences published against oxytetracycline, which is an antibiotic, which according to legislation, the levels of it within milk should be regulated. Okay, so Caitlin, carry on pressing, yeah? Yeah. Once more. And bang. Okay, so for example here, here if you pause a bit, the results here show this characterization where amongst the few groups that actually invest so much effort into this biophysical characterization of the aptamers themselves, which is super important in order not just to identify the best performing candidate sequence, but also get some structural information for the sequences, which is invaluable in setting up, in translating this biorecognition event into something that is usable when it comes to the fabrication of the sensor. So you want, perhaps a lot of times, these aptamers, when they interact with their target, there's nothing changing. It's like two bodies touching one another. This obviously won't work for the fabrication of a sensor. And so our job is not only to characterize this binding, how strong this binding, this interaction is, but also to engineer the aptamers in such a way so that this biorecognition event gets translated into a measurable signal. And that's another big advantage of aptamers versus antibodies, that being nucleic acids, their sequences can be easily truncated, mutated, and so on to suit the needs of your assay. Okay, so what it shows here, what's shown here is all these, how we used all these biophysical techniques to select and then engineer the sequences in such a way so that we achieve our goals. So if you carry on, Caitlin. Mm-hmm. Yeah. Yeah. And then for the actual implementation, as I said, the development of the choice, the selection of aptamers, and then their integration into assays and biosensor formats is not straightforward. So once you have your aptamer selected in solution, all this biophysical characterization takes place in solution, then there are different ways that you can integrate your aptamer into an assay. So we tested out with some model aptamers that were of interest to the project, to the NextGenMicrofluidics, different ways of taking these aptamers and putting them on the surface, basically, and getting some quantifiable signal, in our case, fluorescence, if you remember, TERF, out of the biorecognition event. So having examined lots of different, and Kate can carry on pressing now, lots of different approaches, we have concluded on the best one. Here is, perhaps this is of interest, it shows these microarrays spotted on the surface, and you can see that even the spots, with the spotter that Jörg talked about, you can see that even the spots, depending on the material that is being spotted, differ. Okay, so if you carry on pressing, yeah. Okay, this just illustrates the point that I'm trying to make. Carry on. Okay, so the best approach is this one. Again, lots of different ways to, even when you choose the approach, then there is different ways to implement that. So without wanting to tire you, carry on. Yeah. What this shows is this, right? So now we know, it's okay, Caitlin, you don't need to go back. Now we know how to, we had the opportunity within the project to actually use these specific analytes that we wanted to detect it, to develop and put to the test a process flow which allows us to fastly for the speedy development of an assay based on aptamers for the detection of small molecules in specific. And that is of great importance because these are the toughest analytes to be detected, exactly because of their low molecular weight. However, they are hugely important, especially for the food industry. They are the most important analytes, but for other fields as well, medicine, environmental monitoring, and so on and so forth. So again, light sample pretreatment is the holy grail of the of processes that one would like to attain and transfer on cartridge. When it comes to the detection of different biomarkers, different analytes of significance to environmental diagnostics, medical diagnostics, and food quality monitoring, these small molecule analytes are the toughest to actually detect. Aptamers, because of their low molecular weight, they are the best biorecognition molecule against them. But even when you're working with the best probe for the detection of the toughest, nothing is straightforward, right? However, what we managed to achieve within the project is to actually test different ways of integrating all these different scenarios, and now we have a process flow that allows us to develop assays for any analyte with aptamers very fast. Okay, you can skip this slide. This talks about sample pretreatment as a whole, and then we can go to the last slide, basically, which this has to do with the validation of this multiplex detection with a spotted microarray and the detection of two different analytes. There aren't any other cartridges that do that. Some sensor functionalization, which we also developed within the project. I think if we can go to the last slide, yeah, this one, okay, which basically talks about the services, the expertise the partners brought to the demo case and how this was employed within the demo case to develop these in a very short period of time, these two very different types of assays, which, however, share a lot of things in common, as you saw, because if you strip them down to the basics, they both have to do with some sort of sample pretreatment, either amplification, extraction, or whatever, and then the detection of the analyte or analytes from a spotted microarray. So that's the basic concept. Although they look very different, they're very similar at the same time. So the partners that work together in this, in the development of this lab-on-a-cartridge is self-contained, very integrated, yet versatile lab-on-a-cartridge. Had some expertise, but within the project, this expertise was further solidified, and if you want, the connections were established so that we offer within the context of the MIH, the company that was set up that Martin talked to you about, these services this expertise as a service. Okay, so within the project we acquired some new know-how. And we developed some new services, which you see here. For example, I've already talked about the aptamer-based assays. Equally important, the surface functionalization techniques that were developed within the project find a lot of different applications, and they have already been employed in a number of these open call projects for tailoring the surface of the surface properties of different cartridges, so they don't have to be necessarily be employed for the fabrication of biosensors, but they can change. For example, they can be employed, and they have been employed to change the wettability of surfaces. Also, equally important, we got to compare different ways to fabricate the sensor foil with roll-to-roll techniques, and with fantastic results obtained with all of them. Yeah, that's about it. Yeah.
Great. Thank you so much,
And thanks
George, for
your
modifying
attention.
your presentation. And thank you for everybody for your patience with the technical issues. And thank you to Jörg Nestler from Biflow Systems for stepping in at the beginning with the presentation. So we have about 2 more minutes left, and I think we have a question from Saloni Agarwal coming in. Saloni, would you like to unmute and read your question?
Yeah, of course. Really great talks by both of you. And my first question is, what is the sample pre-treatment technique that you employed for PCR methods within the cartridge? Because again, you need a pure nucleic acid for PCR or amplification.
Yeah, so maybe I can answer it. So what we used is like a one-step purification step using special, say, matrix solution from a company, so commercially available solution, which is normally used in special single-step spin columns, which we employed in the cartridge.
Okay, and this would be integrated into the cartridge itself?
Yes, yes.
And what was the sample complexity that you tried? I mean, was it just like swap samples, or was it something more complex? Okay.
It was
And
normal. Perhaps I can. It was we validated the device with a lot of samples collected during the pandemic
yeah.
in Greece, most of them, where the research institute that I work at is based, because we were one of the reference centers for the collection of different samples. So yeah. The samples were already characterized with normal, the standard conventional PCR techniques, the certified kits, and so on. So we had a really good idea as to what the samples were when it comes to the viral load and so on.
Okay. And the last question was, what was the overall sample to result analysis time for this cartridge system?
Yeah, so I mean the longest time is still the PCR, and it also takes some while for hybridization. So it was below 1 hour, so not comparable with the real-time solutions at that time out there in the COVID pandemic, which were targeting basically one parameter, which was not the intention here, as this is a microarray approach where you basically do an endpoint PCR and then do a hybridization. But the main focus was the discrimination of different strains and different viruses. So it's a little bit below 1 hour.
Okay, yeah. Great work.
Great, thank you, Saloni, for your question.
Thank you.
Thank you.
Okay, any last questions? We had one about sending the presentations, so we'll see if we can put together shareable presentations and send to any.
And send you the one that was meant to be
the real
presented.
presentation. As the little bonus, you'll get an email afterwards. Great. Well, I put in a link to our next webinar in the series. Will be on February 5th, foil-based microfluidic chips for multiplex in vitro diagnostics. Is it a game changer for lab quality at low cost? So our partners from Ionium Research and GenSpeed Biotech will be presenting their demo case in this webinar. And don't forget to join us if you can in Graz for the Human Health Tech Hub Hysteria event and our event on March 26th. Thank you everybody for coming. Thank you again for our speakers, and we look forward to staying in touch. Have a great day.
Thank you.
Thank you.
Thank you.
Bye-bye.
Have a good day. Bye-bye.
Thank you. Bye-bye.